All right, guys, got my afternoon caffeine source. So let's pick up where we left off in the virus lecture. Also wanted to let you know, I'm going to go into Talon. Now, not a tech-savvy gal right here, but I'm going to try to open up some type of chat. So if there are any questions on the content, they could be posted there and I could answer them. I guarantee you that if you have a question, there's going to be other students in the class that have that same question. So I'd love to do it in one place where everyone can see all of the answers. So let me work on that. I may have to reach out to our Kirkwood Talon guy, but luckily he's really responsive. So hopefully I'll get that organized soon. Okay. So last time I talked about acute viruses such as, influenza, hepatitis A, where you get sick right away. But then the good news is the virus is cleared from your body. The other virus, that's Eileen, my cat, my three-legged cat, whining, just so you know. The other type of virus, like HIV, are what we call latent viruses, and they tend to produce chronic diseases that can come and show up again and again and again, like shingles with varicella zoster, or getting cold sores again and again and again from herpes simplex 1. Okay, those are examples of latent viruses. So what I want to do now is go through the steps of how viruses multiply inside a cell. And then I want to tell you the difference between acute or lytic viruses and chronic and latent viruses. Okay, so we'll go through multiplication and I'll define all of those terms. So once again, just as a refresher, remember viruses are obligate intracellular parasites, which means that they cannot replicate unless they are inside a living cell and they basically hijack all of the cell's machinery for themselves. Okay, so you gotta have a host cell to have the virus multiply. So here are the 6 different steps, and I'm gonna go over these one by one. Should you know them in order for an exam? Yes, you should. And hopefully when you learn what they are, the order is gonna be kind of common sense to you. Okay, so we're gonna go through each of these steps in order. Okay, I wanna show this. This is a good overview slide. So basically what we see here is, here you have the virus, and it's coming up to the cell. And in this one, it is an enveloped virus with spikes. Okay, and remember the spikes aid in attachment. So they fit like a lock and key, and the cell is like, well, you must be friendly because the lock fits in the key, so come on in. And then what will happen is the virus basically dissolves, and so just the nucleic acid, which is either RNA or DNA, is present. And then this is where it goes down and uses a cell's machinery. Okay, so it's going to use, in this case, it's an RNA virus. So it's going to use RNA polymerase to make more copies of the RNA. You're going to have translation happen so that you can make new spikes, you can make new capsomeres. Then all of those get put together and then they leave the cell. Okay, the only thing that this animation isn't showing you is that you don't make just one little virus. You're making potentially hundreds of thousands of viruses. Okay, so this is a good overview slide to look at. Okay, so step 1 is adsorption. Now that's not a typo, okay? It's not supposed to be absorption with a B. Absorption is when you have a solid that is soaking up a liquid, okay? So when you clean up a liquid with a paper towel, that's absorption. Adsorption is when a solid meets a solid, okay? And in this case, it's usually those protein spikes that attach to receptor proteins on the host cells, okay? That's what we refer to as adsorption. So that's the first step. Okay, and remember, the key that fits in the lock is super, super specific. So a virus is only going to attack usually a single species of organism and only one type of cell, okay, such as respiratory cells or cells of the immune system. Okay, so here we're just showing, here's the protein spikes. Here is the key fitting into the lock. The cell doesn't know any different. It's like, well, you have a key to my house, so you must be friendly. Come on in. And once again, how those the keys or the spikes develop is simply through random genetic mutation. Okay, there's not any thought process that goes into that. Second step is through penetration, or is called penetration. So this is where the cell is basically welcoming the virus into or inside the cell. And by the time you get to this point, it's kind of too late to do anything because now the virus is inside. Okay, so the way that this usually happens is by the process of endocytosis, and this means cell eating. We talked about this a little bit in chapter 6, how it is, it's an active process, so it requires ATP. That's what we had talked about before. In this case, basically, the cell engulfs the virus and brings it inside. Okay, that's one possibility. The other thing is that if it's an enveloped virus, remember the envelope is made up of a lipid bilayer, and so a lot of times that envelope just fuses directly with the cell membrane, and that's how it gets into the cell. FYI, I just wanted you to meet Eileen, the three-legged cat. Anyway, I digress. Okay, so here's some examples of both of these. So here you have a naked virus, and it attaches via its spikes, and then the cell, because it thinks it's friendly, goes through endocytosis and then just brings it into the cell. Okay, the other example is if you have an enveloped virus, it attaches with its spikes, and then the envelope just fuses with the lipid, and there we go. But the end point is exactly the same. Now the virus is inside the cell. Okay, uncoating is basically the virus getting naked. Okay, so once it's inside the cell, it needs to free up its nucleic acid, whether it's DNA or RNA. And so everything else, the capsid dissolves, the envelope dissolves, the spikes dissolve, and now you have the free nucleic acid of the viral genome inside the cell. Okay, that DNA or RNA is then going to direct the process of synthesis. So if it's a DNA virus, then the DNA is going to take the cell's RNA polymerase and go through transcription, and then that messenger RNA will then go to a ribosome and go through translation. If it's an RNA virus, it doesn't have to worry about transcription because it's already RNA. It just heads straight to the ribosome and starts synthesizing viral proteins. Okay, so yeah, it's basically the viral genome that's driving all of this process. So what's happening is it's making new capsomeres, which can then be put together in a capsid. It's making new spikes. It's synthesizing more of its DNA or RNA. Now you have all of the parts, okay? Now the next step is you put them all together into actual mature virus particles. And then finally, the last step is release. And this can happen a couple of different ways. Okay, so enveloped viruses are really smart. They actually steal part of the cell membrane on their way out. And really what a perfect disguise, because now they look like exactly one of your cells. Now, eventually, some of the proteins in that lipid bilayer are going to become viral proteins. And so eventually, your immune system is going to be able to tell the difference. But, hey, what a great way to disguise itself. Also, it can bud off, or exocytosis, lysis. Basically, the cell just kind of blows up. Okay, so here is the big important number that should freak you out a little bit. Once again, don't remember numbers for a quiz or an exam. I'm not going to ask you anything that nitpicky, but for every one virus that infects a cell, when it goes through viral replication, it's now going to release anywhere from 3,000 up to 100,000 new viruses. And the term that we use is virions, but basically viruses. And guess what? Now those viruses can go and infect other cells. So one cell was infected. Now you potentially have 100,000 of your cells infected. Okay, the entire length of this cycle can be anywhere from 8 to 36 hours. Once again, don't memorize those time frames, but this explains how, you wake up in the morning, you got a little tickle in your throat, and then by the end of the workday, your throat feels like it's on fire. And that's because your cells are lysing, the virus is spreading, and your cells are basically being destroyed. Okay, so yeah, that's gonna not feel good. Some viruses take longer, and this is if you've ever had, say, influenza, and you're like, you know what, I've been feeling rotten, kind of weak for a couple of days, I'm coming on with something. Yep, it's a virus, but it's taking a little bit longer to go through these steps. Okay, but this to me is completely freaky. How can we not all be completely destroyed by viruses when we're producing that many? Well, as we've seen from COVID-19, people do die, okay? Most viruses are non-life-threatening, and there's a couple of reasons for that. So I'm gonna go through that a little later in the PowerPoint. Okay, so here's release. You can either have, basically the cell just kind of blows up, or here we go, I'm gonna steal part of your membrane, and now I've got my own envelope, and it's a perfect disguise. Okay, so how do we tell, in the lab that someone has had a viral infection versus a bacterial infection? One of the things that can be done in the pathology lab or histology, where you're actually looking at sections of tissue, is looking for something called cytopathic effects, or CPE. And this basically states that when cells are infected with a virus, they tend to look very characteristic under the microscope. Okay, they have things called inclusion bodies, which are basically big masses of viruses. They cause like holes in tissues. It's very characteristic, and the cells look different as compared to damage from a bacterial infection or a fungal infection. Remember too, the other reason that we have to look under the microscope for damage is because we can't do a gram stain on a virus and have it show up. You can't look for fungal cells under the microscope. Viruses are so small that if you want to identify them directly, you have to have an electron microscope. And there are very few hospitals that have access to an electron microscope. Okay, so a lot of times it's kind of, let's look at the damaged tissue after the fact and prove that it was a virus. So let me show you what this looks like. Okay, so if we look up here, these are normal cells, okay? And the dark purple things in the middle are nuclei. Now, normally, cells are good neighbors, okay? They communicate with each other. They know when to start reproducing. They know when to stop. They're very orderly. And so you can see this looks pretty uniform. When it's infected with a virus, you get, this is what I'm talking about as inclusion bodies, like you have clumps of nuclei, you can have clumps of viruses, you have big holes in the layers of cells. They just look really weird, okay? So that's one way that, a lab scientist or a pathologist could look under the microscope and say, this was a viral infection, and I can tell because I'm seeing CPE on the cells. Okay, so here is where I'm going to talk about lytic versus latent. Okay, and one thing, and let's see, I'm going to make myself a note. Where's my pen? Because I want to add this to some notes. Okay, so lytic viruses cause acute illnesses. Okay, lytic viruses cause acute illnesses. And what's happening with a lytic cycle, it means that it's going to go through those steps 1 through 6 right away, and you're going to destroy the cell. So yeah, the bad news is you get sick, it happens right away. The good news is it's going to end and it's going to be over, and your immune system is going to clear that virus away. Okay, so examples of this would be influenza, rotavirus, which causes, mostly in little kids, but it causes nausea and vomiting. Adenovirus, which gives us our upper respiratory sore throats, and hepatitis A. Okay, much more insidious is what we call latent viruses. And some other terms that you may hear, lysogenic, lysogeny, that means the exact same thing. And latent viruses cause chronic viral infections. I'm making myself a note so I can put that in class notes. Okay, so some examples here are ones that we talked about before. HIV. Okay, so HIV, a person, if HIV becomes active, they can get full-blown AIDS several times. You know, you don't have AIDS just once and then it goes away. Same thing with hepatitis B and C, and herpes simplex 1 and 2. One of the other examples of a latent virus is varicella zoster, which, remember, first causes chickenpox and then unfortunately goes to migrate in the spinal cord and then can come out again and again and cause shingles. I also want to define different types of viral infections. So some of these we've talked about. Okay, so acute means, yep, once you get the virus, you're going to get sick in probably a couple of days. You're going to be sick maybe up to a week, but then it's going to go away. Okay, so that's kind of the best case scenario. The worst case scenario is chronic, where new viruses are continuously produced at different time intervals, and you just can't get rid of it because the virus is with you. Okay, then you kind of have these terms in the middle. So persistent viruses means it's basically people who have weaker immune systems or are immunocompromised, you know, such as the elderly. It's basically an acute virus, but God, your immune system is weak and you just can't shake it. So unfortunately, you have small amounts of virus and then they come back and they can cause just these, you know, ever-present, persistent symptoms. So persistent, I tend to link that with people who are more immunocompromised. Latent infections is when the viral DNA is inserted into the host DNA, and then there's no symptoms in between, and boom, outbreaks. Okay, so those of you who unfortunately have to deal with herpes simplex 1, you know, maybe you have a big exam coming up, boom, there you have some cold sores. And then it goes away for a couple of months. And then, you know, the next time you have a big stress, boom, here come the cold sores again. That's an example of a latent infection. Okay, let me see if I want to skip this one. No, that's okay. Okay, so actually, let me see how much we have left. You know what? I'm going to stop here because we're almost at the 20-minute mark, and then I'll just have one more short video to do, and then we'll be done with viruses. Thank you, Lord. So anyway, hang in there, guys, and I'm going to get this uploaded, and hopefully it'll get there sooner rather than later. Okay, thank you. We'll pick up on this slide next time.